Hi all, welcome to today's session. The topic is the basics of microbial taxonomy, the concept of species, taxa and the strains. I am Greeshma, Assistant Professor, St. Mary's College, Thrissur. The microbial taxonomy. As the term indicates, the taxonomy. What is taxonomy? The taxonomy is the science of biological classification, which is a biological classification and mainly consisting of three interrelated parts: classification, nomenclature and identification. And because of the wide biodiversity of living organisms, it is very desirable to classify and arrange them into groups based on their similarities. This is done by the taxonomy. Taxonomy is the classification, that is the biological classification, and consisting of three separate but interrelated parts: classification, second one is the nomenclature, and third one is the identification. And moving on to the first, that is the classification. What is classification? The classification is the arrangement of organism into taxa. That means we are arranging the given isolate into a taxa. Taxa is also known as group. That is, this arrangement is based on the mutual similarities or by evolutionary relationships. And so the classification is the arrangement of organism into taxa. The second one is the nomenclature. As the name indicates, that is nomenclature, that is giving names. The assignment of names to taxa. And it's a branch of taxonomy concerned with the assignment of names to the taxonomic group in agreement with the published rules. We are agreeing the published rules for assigning the names to this identified taxa. Then third one is the identification. One, we are classified, then we are given the names, then finally we have to identify it. That means the determination of taxa to which an isolate belongs. And it is the practical side of taxonomy and the process of determining that a particular isolate belongs to a recognized taxa. That means the given isolate belongs to which taxa or not. We are determining it. Then, what is the importance of taxonomy? The taxonomy has got some importance that means to organize a huge amount of knowledge, or the scientists got a huge amount of knowledge based on this taxonomy. Then second one is by using this taxonomy, you can make predictions or frame hypotheses about the organisms. You can make a hypothesis, that means which belongs to this group, that means or another group or not. And third one is it places the organisms into a meaningful or useful groups, and it helps to communicate between the scientists. And last one is essential for the accurate identification of organisms. It helps for the accurate identification of organisms. So the first one, it will provide huge amount of knowledge to scientists. Second one, make predictions or frame hypothesis about the given isolate. Then third one, it will places the organism into meaningful, useful group. And fourth one is accurate identification of organisms. And now we are moving to the next slide. The ranks or levels of bacterial taxonomy. That means based on this classification and all those things, certain ranks or levels will be assigned. That means mainly 7 levels. That means the highest level is the domain and lowest level is the species. The species, then followed by the genus, then family, then order, class, phylum. Finally, the highest level, domain or kingdom. The species is the basic taxonomic group in bacterial taxonomy, and certain groups of the species together form the genus or genera. Then the groups of genera are collected into families or family, and these families are collected to form the orders collected to form classes, then classes together form the phylum or phyla, then finally the domain or kingdom. For example, we can say about the bacteria. The bacteria is the domain. Its phylum is known as Proteobacteria, and its class is the alpha Proteobacteria, the order Enterobacteriaceae, then the family Enterobacteriaceae, then the genus is the Escherichia, then finally the lower level species Escherichia coli. So coli is the species, Escherichia is the E. coli. So this is the level, the taxonomic hierarchy: species, genus, family, order, classes, phylum, and the kingdom. And moving on to the history of the taxonomy. For the scientist Carolus Linnaeus, he is a Swedish scientist, and he is considered as the father of taxonomy in 17th. And he contributed main two contributions: the hierarchical classification system, and the second one is the binomial nomenclature. This hierarchical classification system, as we discussed earlier, and next one is the binomial nomenclature. As the name indicates, this all you know, the two genus and the species. The genus which is capitalized and it is italicized, and the species which is italicized but it is written in small letters. And based on the hierarchy classification system, the kingdom, that is divided into, according to Carolus Linnaeus, it is divided into animals, plants, and the minerals. And there are mainly different kingdom classification system: 2 kingdom classification, then 3 kingdom classification, 5 kingdom, 6 kingdom, then finally 8 kingdom classifications. And first one is the 2 kingdom classification. The 2 kingdom classification is proposed by the scientist, the father of taxonomy, Carolus Linnaeus, in 1758. And he classified into the kingdom into 2: kingdom Plantae and kingdom Animaliae. And the second one is the 3 kingdom classification. And this kingdom classification is proposed by the scientist Carl Woese in 1977. And he divided the cellular life forms into 3, as it is known as 3 kingdom. The kingdom Archaea, then kingdom Bacteria, then kingdom Eukaryote. This Archaea and Bacteria is a subdivision of prokaryotes. Okay, Archaebacteria and the Eubacteria is a division of prokaryotes. The bacteria is also known as Eubacteria. The Archaea is also known as Archaebacteria. And this classification is mainly based on the 16S rRNA gene difference. 16S rRNA difference. He classified the whole cellular life forms into 3: kingdom Archaea, kingdom Eubacteria, then kingdom Eukaryote. This is the 3 kingdom classification. And next one is the 5 kingdom classification. And it is proposed by the scientist Robert Whittaker in the year 1969. And he used mainly 3 criteria for their 5 kingdom classification. That is the cell type. Then second one is the level of organization. And third one is the nutritional type. Cell type means whether it is prokaryote or eukaryote. Level of organization means it is unicellular or multicellular. The nutritional type means mainly 3 types of nutrition, that is photosynthesis, absorption, and ingestion. And based on these nutritional type, based on these three criteria, that is cell type, level of organization, nutritional type, classified the life forms into 5. And another important factor is that he given a position to the fungi in this classification. And it is the fungus that is not well established, and it is oscillating between the kingdoms Protista and the Plantae. So he given a position in this 5 kingdom classification, and he divided into the life forms into 5. The kingdom Plantae, kingdom Animaliae, kingdom Fungi, kingdom Protist, and the kingdom Bacteria. Or otherwise it is known as kingdom Monera. This bacteria is also known as kingdom Monera. And next, this 5 kingdom classification that is not accepted by many of the microbiologists, and a major problem is its lack of distinction between Archaebacteria, Eubacteria, and all those things. So he classified, so the 6 kingdom classification. So in the 6 kingdom classification, the simplest option to divide the kingdom Monera into 2, kingdom Eubacteria and the kingdom Archaebacteria. Kingdom Archaebacteria. The kingdom Monera in the 5 kingdom classification is divided into the kingdom Eubacteria and the kingdom Archaebacteria. So 6 kingdom. The kingdom Plantae, kingdom Animaliae, kingdom Fungi, kingdom Protista, then kingdom Eubacteria and the kingdom Archaebacteria. And these are also based on the cell type and the cell type and the level of organization, whether it is unicellular or multicellular, or whether it is prokaryotic or eukaryotic. And the last classification is the 8 kingdom classification. This 8 kingdom classification, it is proposed by the scientist Cavalier Smith, and in this classification, the bacteria are divided into further forms, mainly 8 mainly 8 kingdoms in the same kingdom, that is kingdom Plantae, then kingdom Chromista, then kingdom Fungi, then kingdom Animaliae, then kingdom Protozoa, then kingdom Archaeozoa, then kingdom Eubacteria and kingdom Archaebacteria. And these are all about the kingdoms. And so there are 2 kingdom classification, 3 kingdom classification, 5 kingdom classification, 6 kingdom classification, and finally 8 kingdom classifications. And all this taxonomic hierarchy and all those, important one is the species. The species. What is species? It is the basic and the most important taxonomic group in the bacterial systematics, or the microbial taxonomy. The species is the basic and most important taxonomic group. And the term species, that is the term species, the level of, as we consider the level of bacteria, it has some certain distinguishing features, and that are generally has some close resemblance to one another, and it is essential for the organization. So it is the term species is applied to the bacteria, and it is a distinct group of strains and has some distinguished features and a close resemblance to one another. The species is that has close resemblance to one another. And another term is the subspecies. What is subspecies? A species may be further divided into 2 or more subspecies. Species may be divided into 2 or more subspecies based on the phenotypic variation or the genetic differentiation. Based on the phenotypic and the genetic variations, species may be subdivided into 2 or more subspecies. And the subspecies is the very lowest taxonomic rank. This is about the species. And next one is the strains. Next term is the strains. What is strain? The strain is a single organism or pure culture isolate. Strain may be the single organism or pure culture isolate. And these strains may be vary from each other in many ways. And some may be biovars. What is biovars? That means they may differ biochemically or physiologically. It is known as biovars. The strains which are biovars means they differ biochemically and physiologically. Then some may be morphovars. What is morphovars? As the term morpho, that is differ morphologically. And the third one is the serovars. That means that it may differ in the antigenic properties. So the strains, it's a single organisms and distinguishable from other within a taxon. Population of organism that is distinguishable from others within a taxon. And another term is the type strain. That is usually one of the first strain of a species study. The first strain of a species studies is known as type strain, and often most often fully characterized and not necessarily most representative member of the species. So the type strain is the first strain of a species studied. So strain is the single or group organism or pure culture of isolate, and that may be very biovar, morphovar and serovar, and the type strain is also noted. And next is the classification system. There are certain classification system, that is natural classification system, that arrange the organism into groups based on the many characteristics. And two methods for construction, that is phonetically and phylogenetically. Phonetically means based on the overall similarities. Then phylogenetically means purely based on the evolutionary relationship. Phonetically and phonetic classification and phylogenetic classification. What is phonetic classification? Phonetic classification is based on the mutual similarities. Phylogenetic classification, it is also known as phyletic classification. This all discussed later in the following sessions. Next is the phonetic classification is based on the mutual similarities. Then phylogenetic classification is based on the evolutionary development of a species. And another criteria is the major characteristics. What are the characteristics used for this taxonomy, for the classification? Okay, then the two major type of characteristics, classical characteristics and molecular characteristics. Okay, the classical characteristic means mainly four categories: morphological, physiological and metabolic, ecological and genetic. Okay, the classical characteristic used for the taxonomy is the classical. What is classical? The first characteristic is the morphological characteristics. It is very simple, that is based on the cell size, cell shape, then cell structures, then cell arrangement, then cell color, then staining, the motility difference, spore formation, all that we are characterizing. Next, second one is the physiological and metabolic. As the name indicates, it is based on the carbon nitrogen sources requirement, then the energy sources, then cell constituents, the temperature, pH and all those we are based on all those we are categorizing. Then based on the ecology characteristics, ecological characteristics based on the life cycle pattern, then the symbiotic relationship, then ability to cause disease, whether it is a pathogen or not, and based on the growth requirements, all those we are characterizing. And next one is the genetic analysis, is also a classical classification. Genetic analysis means based on the transformation or conjugation, then the chromosome gene exchange, study of chromosome gene exchange, all those. These are all about the classical characteristics. The second one is the molecular characteristics. Molecular characteristics, as the name indicates, the molecular levels. That means based on the proteins, the the nucleic acids, whether the RNA and the DNA. First one, it is based on the molecular characteristics is based on the comparison of proteins. Then second one is the nucleic acid base composition. Then third one is the nucleic acid hybridization. Then finally the nucleic acid sequencing. These are the four molecular characteristics used for the taxonomy. This classification, classical characteristics and the molecular characteristics will discuss in the following sessions. I think all these portions are clear, and if you have any doubt, please ask me in the discussion time. Okay.